Hello class. So this is the second to the last topic that we have for the end term. So this is a discussion which is also connected, or should I say associated to the previous topic, which is the restriction enzymes. Okay, because in the topic of restriction enzymes we learned that a plasmid or plasmids are used as vectors. Okay, so for this discussion we will mainly focus on number one, the structure of the plasmid. Okay, the basic characteristics of the plasmids, and of course the uses and applications of these plasmids. Okay, so as what you can see here in the illustration, okay, the main or the common shape of the plasmid is what again? Okay, they are circular DNA. Okay, and again, let me reiterate that plasmids are usually double stranded. And aside from that, plasmids are also considered as replicons. This one. Okay, replicons. Okay, so meaning to say they are units of DNA capable of replicating. That's why they are called replicons. And they can replicate autonomously. Okay, autonomous replication, especially if they are already transferred within the host. Again, as what I have said, plasmids are considered replicons, meaning to say they are units of DNA capable of replicating autonomously within a suitable host. Okay? And of course, I know that you already have your topic in virology, right? Virology. So I think you already have an idea of what a virus is, and of course its structure. Okay, I am mentioning that one because unlike the viruses, okay, the viruses class, they are encased. Okay, their genetic material is encased in a protective protein coat, right? So if this is the virus, for example, this is the genetic material of the virus. Okay, so they have this protein coat, okay, surrounding them. This one. Oops. Okay, this one class. So let's say, for example, my pencil is not working. Okay, for a while. So let's say you have there the genetic material of the virus, and then of course they are surrounded by a protein coat. This one. Okay, so they are surrounded by a protein coat. I think I have illustrated that one in. Okay, let me rephrase it again. Okay, redraw it again. So this is the genetic material of the virus. And then again, there is a protein coat covering, okay, that genetic material. So you have here, okay, what do you call that one? Okay, the one covering the genetic material of the virus. I know that you have learned that in your virology. Okay, they are made up of these basic subunits. That's circular. Okay, those are capsomers class, and all together they make what? Okay, they make a capsid. Okay, capsid. So that is the one which covers the genetic material of the virus, right? Okay, so I am mentioning this one again because unlike again the viruses, the plasmids are considered as naked DNA. Okay, naked DNA. Okay, and of course, unlike your other microorganisms, okay, the plasmids do not encode genes necessary to encase or to enclose the genetic material, okay, for transfer to a new host. So that's also one of the differences between the genetic material of a virus and the genetic material of a plasmid. Okay? However, as also mentioned before, okay, in our previous discussion, some of the plasmids, okay, some classes of the plasmids might encode the conjugative sex pilus, right? So if one bacterium has this sex pilus and the other one has sex pilus, of course, okay, they can transfer the genetic material through this structure, okay? So that's, again, okay, some infos about plasmids. Okay, next one. Okay, I think I have shown you this in the previous discussion. Okay, as what you can see there, the green structure, that's the bacterial cell. Okay, and labeled here is the bacterial DNA. and you have the plasmids. Okay? So in short, plasmids are separate from the bacterial chromosomes and they can also replicate independently. That's why again and again we are referring the plasmids as extra chromosomal DNA. Okay? And aside from that also, you have to take note that plasmids generally carry only a small number of genes. Okay? And in the laboratory and in the field of medical technology, these genes class are notably associated with what type of characteristic, okay, of the bacteria. Okay, they are commonly associated to antibiotic resistance. Okay? And another one is what you can see there again and again, they are mostly commonly found where? Okay? In the bacteria, in the prokaryotes. However, you have to take note that they can also be present in the archaeans and other eukaryotic microorganisms. Okay? And aside from that, again, they are small and they are also circular and they are double-stranded. Okay, I'm repeating that one so that you will not forget the characteristics of the plasmid because you need these basic things, okay, of our topic in order for you to understand the more complex information that will be presented to you in the future. Okay, another one. Okay, again, plasmids are extra chromosomal DNA. Okay, DNA duplexes that carry genetic information, and although they have only that small number of genetic information, usually that genetic information that they carry is very useful. That's why plasmids are usually used, okay, in genetic cloning, in other applications like in gene therapy also. So because plasmids are easy to manipulate also, and of course because of their small size, okay, they can be easily used in the laboratory. Okay, DNA helices can assume both linear and circular form. Okay, however, as what I have mentioned, usually class bacteria are having this circular plasmid. Okay, and most plasmids again are double-stranded and circular, and they only have 2,000 to 100,000 base pairs. So that is equivalent to 2 to 100 kilobase pairs in size. Okay, that's their size. And the plasmid DNA duplex is compacted or supercoiled. Okay, so what is mentioned here is they are extra chromosomal, right? So just like your chromosomes class, you have to take note, okay, to summarize this information. Plasmids also carry genetic information, okay, and due to their size and effect, of course, okay, on the host cells, for examples, okay, plasmids can only carry a limited number of information, okay, but although limited, but these information coming from their genes are very useful. Okay, so speaking of the supercoiled plasmid, so you have here, okay, so that is a supercoiled DNA or the supercoiled plasmid. So again, that supercoiled plasmids, okay, can be relaxed by nicking, okay, meaning to say breaking of one strand, okay, breaking the plasmid duplex, okay. So what will happen? They will be relaxed. So this is the relaxed form, okay. Or if you break two strands, it will result to the linear form, okay. Again, if you nick or you break only one strand of the plasmid duplex, okay, it will be a relaxed form or a relaxed circle. But if the supercoiled plasmid, okay, if both of the strands are broken, then they will lead to linear plasmid. Okay, and another thing, okay, we have also discussed about electrophoresis, right? Because the different physical states of the plasmid DNA class can be resolved by distinct migration characteristics during gel electrophoresis. Okay, and another thing, okay, Before I go deeper to other topics related to this one, especially the cloning vectors, okay, I have mentioned about antibiotic resistance. Okay, and these are the mechanisms to which the bacteria can acquire resistance. Okay, when we say again susceptible bacteria, meaning to say they can easily be killed or eliminated by antimicrobials, right, or antibacterial. Okay, so if they are susceptible, okay, and they infect a patient, of course, the patient might heal after administering antibiotic to that patient. However, if that susceptible bacteria acquire resistance, okay, to antimicrobials, then the previously used antimicrobial can no longer be used to these kinds of bacteria. Okay, so how does a bacterium, okay, how does a bacterium acquire resistance? So number one, through genetic mutation. Okay, I know you are aware of this already. Number two, accepting antimicrobial resistant genes from other bacteria. So that is what we call generally as genetic transfer. Another thing, so that resistance usually occurs through this biochemical mechanism. So number one, destruction or inactivation. So this is just a very simple process in which antimicrobials are used, okay, to destroy the bacteria. Okay, number two, efflux, and then number three, again, mutation. Okay, so let's have first the destruction or activation. So many bacteria class possess genes which produce enzymes. Okay, so you take note that in destruction or inactivation, enzymes are involved here. Okay, that chemically degrade or deactivate the antimicrobial, rendering them ineffective against the bacterium. So by the use of this antimicrobial or antibacterial, then of course, we might kill the offending pathogen. So let's say, for example, this one. So this red structure here are the antimicrobials. So how do they perform their action? So of course, when we administer antimicrobial, okay, they will enter the bacterial cell. Okay, and of course, again, some of the bacteria might acquire resistance. And again, in destruction and inactivation, what are involved here? Okay, the enzymes, right? So, if the bacteria are resistant to antimicrobial, meaning to say they might have enzymes that can degrade or modify, okay, the activity of antimicrobials. Okay, so meaning to say the antimicrobials will be rendered ineffective before they can even reach, okay, the target site, and of course, okay, before they damage the bacterial cell. So that is simply destruction and inactivation, usually with the aid of the enzymes that destroy the antimicrobial agents. Okay, how about this one? This is efflux. Okay, so you have here the structures, okay, which are innate in a bacterial cell. Okay, in some of the bacteria, so you have the porine channel and of course the efflux. Okay, so the efflux pump class is a protein channel that actively exports antimicrobial and of course other compounds out of the cell. So this is a bacterial cell. Okay, so this one class, this is a bacterial cell. Okay, another thing. So if ever an antimicrobial is administered, for example, so that red ones are the antimicrobials, okay, they can enter the bacterium through the porine channel. But what do you notice here? Okay, the efflux pump class, they pump out, okay, the antimicrobials out of the bacterial cell. So meaning to say, okay, that's one way of the bacteria in eliminating the, what you call this, the antimicrobials. Okay, by actively pumping antimicrobials, okay, class, the efflux pump, okay, the efflux pump prevents the intracellular accumulation necessary to exert their lethal, okay, lethal activity inside the bacterial cell. So efflux pump, number one, destruction or inactivation, of course, with the aid of the enzymes that destroy the bacterial cell. Number two, we have the efflux. Again, by means of efflux, we have the efflux pump, which will, again, pump out the antimicrobials, okay, out from the bacterial cell to render it ineffective. And of course, you know this one, mutation. So mutation is a change in the DNA that can sometimes cause a change in the gene product, which is the target of the antimicrobial. Okay? So for example, again, these are my antimicrobials. So when a susceptible bacterium class comes into contact with a therapeutic concentration of antimicrobials, like let's say we have ciprofloxacin, or let's say we have norfloxacin, okay, whatever antimicrobials you are thinking there, okay? So the antimicrobials can bind to a specific site. For example, the antimicrobial can bind to, let's say, DNA gyrase. Okay, others call it as gyrase. Okay, others call it gyrase. Okay, DNA gyrase. So you have to remember that this enzyme class is necessary for DNA replication. So of course, without DNA gyrase, okay, the bacteria cannot replicate. So sometimes the antimicrobials will bind to this enzyme to render the, I mean, some of the antimicrobials will bind to these enzymes in order to stop the DNA replication. So, of course, the bacteria will not multiply. Okay, so meaning to say that antibiotic can stop DNA replication of the bacterial cell and stop its multiplication. So, just like this one class. Okay, once the antimicrobial enters the cell, it can bind to the specific site. Let's say this is a ribosome, okay, which is necessary for protein synthesis. So if the antimicrobial blocks the ribosome, then the bacteria cannot create proteins necessary for its function. Like for example, okay, DNA replication. Okay? So that is a type of mutation. Okay? Another one, why is it a type of mutation? Okay, it will only be a type of mutation if there are mutations, okay, in the structures where the antimicrobials are bound to. Okay, let's say for example this one. Okay, so what if this one has mutated? Sorry, let me go back to this one. Okay, what if this one has mutated, that instead of having that site to bind here, it will become like this. The ribosomes, for example, mutated, okay, in a bacterial cell. So the antimicrobial can no longer bind to that specific structure. So can the antimicrobial be effective in killing or eliminating the bacteria? Of course not. So that is one way of course, okay, stopping the replication, the multiplication of the bacterial cell. Okay? Another thing, we have genetic transfer. Okay? Genetic transfer, I think I have already illustrated one last time, the conjugation. But aside from conjugation, we have other types of genetic transfer. We have conjugation, we have also transformation, and we have also transduction. So the genetic material can be transferred between bacteria by several means: conjugation, transformation, and transduction. So number one, we have conjugation. So conjugation is mediated by a plasmid. Okay? It's the plasmid again that will replicate independently. Right? They can replicate autonomously, as what I have mentioned earlier. Okay? Independently of the chromosome. So this one is what you can see there, the structure that is a sex pilus. So the plasmid class usually again, okay, may help, okay, the bacteria in adapting to its surroundings, because these plasmids might carry genes that can confer resistance to antimicrobials. Example, when two cells are in close proximity to each other, just like in the illustration here, okay, so there will be a formation of this sex pilus. It's like a hollow bridge, okay, or a bridge-like structure, okay, that will form between the cells, especially if they are close to each other. And in that case, it will allow a copy of the plasmid, okay, as it is duplicated, to be transferred from one bacterium to another. So if this one is a susceptible bacterium and if this one is a resistant bacterium to a certain antimicrobial, then that green structure there transferred to the pink bacterium, which is susceptible, okay, will make the pink bacterium be resistant to a certain antimicrobial agent. Okay, that's simply conjugation with the help of sex pilus. Okay, how about this one? Transformation. Transformation. So you have to take note, class. Okay, the clue here is this process, okay, the transformation. What is happening here is that the genes are transferred from one bacterium to another as naked DNA. When you say naked DNA, these are DNA class which are not associated with proteins, with lipids, or any other molecules that protect it. And usually, naked DNA are histone-free DNA. Okay? And another thing, these naked DNA's class, usually they just float in the cytoplasm of the cell, okay, rather than being enclosed in a nuclear membrane. And of course, one of the examples of the microorganisms having this naked DNA is again the bacterial cell or other prokaryotic cells. Okay, so transformation. So for example, in this illustration, you see two bacteria. So this one and this one. Okay, the green rings here, class, is what you can see there. Okay, those are resistant genes. Okay, those are plasmids again. I mean, carrying the resistance gene. So the green one, that's a plasmid of one bacterial cell. And this red one, class, that's again the resistant gene carried by that plasmid. So you have several there. Okay, so when two bacteria are close to each other and one of the cells die or break apart, okay, the DNA's class, excuse me, will be released to the surrounding environment, right? And the other bacteria in close proximity to this DNA, okay, will scavenge this free-floating DNA. So it will scavenge it, and of course, it will incorporate it into their own DNA so that it can acquire resistance. Okay, so meaning to say it's beneficial to the recipient cells. Okay, that is transformation. Okay, the genes are transferred from one bacterium to another as naked DNA. How about this one? You are very familiar of this one, and some of you are even using this in your researches maybe, and this is a common topic in bacteriology. Okay, so in this process, bacteria or bacterial DNA is transferred from one bacterium to another inside what? Inside a bacteriophage. Again, class, a bacteriophage is a virus that infect bacteria. Okay? So, unlike conjugation and transformation, class, transduction does not require physical contact between the cells donating the DNA and of course the cells receiving the DNA. So you can observe that again in conjugation, but in transduction, there is no requirement of physical contact between the two cells. Okay, so what is happening here is that, okay, the bacteriophage class can be used to carry the bacterial DNA so that the bacterial DNA will be multiplied or will be replicated. Okay, so meaning to say, to make it simple, okay, this transduction class is a process whereby a foreign DNA is introduced into another cell via what vector? What type of vector? Okay, very good. Viral vector in the form of bacteriophage. Okay, the transfer of the bacterial DNA from one bacterium to another via a viral vector. Okay, next one. So this one, class, you have here the bacteriophage. Sorry for that. Okay, you have here the bacteriophage, of course, and inside of that is the DNA of the bacteriophage, right? So take note of the color red. Okay, so once the bacteriophage, okay, infect the bacteria, okay, or we can artificially use the bacteriophage to infect bacteria. Okay, later you will realize why. Okay, once the bacteriophage infect the bacteria, okay, the phage, their class, will inject its DNA, right? So once it injects its DNA, okay, into the host cell, what will happen now? Okay, next one. Okay, the structure of the phage will be broken, but the genetic material will be already, okay, found internally, okay, in a bacterial cell. another one. So there will be phage enzymes class that will be produced, and what will happen, okay, as what you can notice, okay, the blue DNA, going back to the first picture, okay, this is the whole structure of the bacterial DNA, right? Take note of the color again, as what I have said. The blue one is the bacterial DNA, the red one is the phage DNA. But what happens right after bacteriophage infection of the bacteria, okay, the phage enzyme will be produced. Okay, the phage enzyme. And that phage enzyme will break down what? Will break down the host DNA or the bacterial DNA into smaller fragments. And of course, what will happen? Okay, the phage DNA class will be replicated, and of course, the phage coat proteins are produced. So that proteins now will wrap the DNA of the bacteriophage, okay, and create new bacteriophage structure. However, we are talking about transduction here. So during formation of these mature phage particles class, the coat proteins, okay, some of the phages, instead of surrounding the fragments of the phage DNA, they may surround the fragments of the bacterial DNA. So both the phage DNA and bacterial DNA's class are being wrapped by these phage coat proteins. So I am emphasizing this one because this one class, okay, this one, the bacteriophage, the coat protein already carrying the bacterial DNA, okay, it can infect another bacteria. So meaning to say, the bacterial DNA can be delivered to another bacteria through a viral vector or through a bacteriophage. Okay? So next one. So once the bacteria will release, once it lyses, okay, all the viruses produced, okay, will infect another bacterium. And it can inject its genetic material. So as what I have said, transferring now the bacterial DNA to another bacterial cell or to another new host. Okay? Just like this one, it's no longer carrying the phage DNA, but it's already carrying the bacterial DNA. So it happens because initially the bacteriophage was used, okay, in infecting the bacteria, so that, okay, the bacterial DNA will be broken into fragments, so that the phage coat proteins will be produced and will wrap both the phage DNA and, okay, the bacterial DNA. So that bacterial DNA enclosed in a viral vector can now be transferred to another bacterial cell. And of course, okay, it will inject again its bacteria. It will incorporate its DNA now to the DNA of the bacterial cell. Okay, because if you are using the phage DNA, it cannot incorporate to the bacterial DNA, right? So once it is already incorporated, okay, the bacteria can then undergo binary fission and they can multiply. Okay, and if this gene is carrying antibiotic resistance, then this one can also carry antibiotic resistance. So that is transduction. So I hope I'm making things clear. Okay, if you have any question, you can just go back to your notes. You can again listen to this discussion, or maybe you can message me. Okay? So speaking of cloning vectors, because it was mentioned already here in bacteriophages in transduction, okay, these cloning vectors are used as what? Okay, as vehicle for transporting foreign genetic material into another cells. So what are the types of vectors that we have mentioned in earlier? Okay, we have number one, the phages, okay, the bacteriophages or the viral vector, or we can make use again of plasmid. Okay, this foreign segment of DNA is replicated and expressed using the machinery of the host organism. Okay, and a cloning vector class facilitates amplification again. Okay, I am emphasizing that because I think I have mentioned that again when we are talking about PCR, another type of amplification reaction. Okay, a cloning vector facilitates amplification of a single copy DNA molecule into many copies. Okay? Another thing, okay, why is it that we are commonly using artificial plasmids? Okay, in the laboratory, they are widely used class as vectors, especially in molecular cloning. Okay, especially of their especially because of their characteristics. Okay, they can transfer the genetic information to another host. Okay, and aside from the fact that they are small and the genes that they carry, okay, are also having small number of nitrogen based sequences, so they can be easily manipulated. Okay, or they can be transferred, okay, through transformation, or they can be transferred through conjugation. Okay, another thing, a cloning vector is a small piece, just like in the plasmid, it's a small piece of DNA that can be stably maintained in an organism and into which a foreign DNA fragment can be inserted for cloning purposes. Okay, the cloning vector may be DNA taken from, again, a virus, the cell of a higher organism, or it may be a plasmid of a bacterium. Okay, and what's again the most commonly used bacterial cloning vectors? Okay, the plasmids. Okay, so in other countries class, I don't know in the Philippines, I think the plasmids, which are synthetic, are available for procurement, especially if you search in the internet. Okay, so they are commonly used in researches or again in molecular cloning. Okay, in gene therapy also they can be used in that instances. Okay, these cloning vectors contain a site that allows DNA fragments to be inserted. For example, a multiple cloning site or polylinker which has several commonly used restriction sites to which the DNA fragments may be ligated. Okay, next one. Plasmids were discovered to be the source of resistant phenotypes in multi-drug resistant bacteria. Okay, so that's, I think, the most common use of plasmids, okay, and MDRs, okay, and also antibiotic resistance, okay, you can hear it often. The demonstration that multiple drug resistance in bacteria can be eliminated by treatment with acridine dyes was the first indication of the episomal plasmid nature of resistance factor, similar to the F factor in conjugation. Okay, what is this R factors class? Okay, because I have mentioned the F factor, we also have here the R factors. Okay, the plasmids which carry genes for inactivation or circumvention of antibiotic action were called resistance transfer factor or the R factors. So these R factors class carry resistance to common antibiotics like the chloramphenicol, like the tetracycline, what else? Okay, ampicillin, streptomycin. And of course, you have to take note that plasmids can replicate in the host cell, but of course, its one drawback is it cannot survive outside of the cell, unlike your viruses. Okay, that's why the acquisition of resistant genes or the resistance genes, okay, from the host chromosomes of unknown bacteria is the presumed origin of this resistance factor of the plasmids. Okay, so again, plasmids can replicate in the host cell but cannot survive outside of the cell as viruses do. Okay, that's why, again, okay, the resistance genes is what I am telling you, okay, from the host chromosomes, okay, transferred to another bacteria. Okay, usually, that's the that's the realization of acquiring resistance, okay, through plasmids. Okay, next one. The circular nature of R factors was demonstrated by buoyant density centrifugation, and plasmid DNA has a density higher than that of the host chromosome and can be isolated from separate or satellite bands in the gradient, of course, also in electrophoresis. Examination of the fractions of the higher density DNA revealed small circular species. The circles were absent from drug-sensitive bacteria. Okay, Compared with fragments of DNA, plasmids are more efficient vehicles for transfer of genes from one cell to another. And upon lysis, the supercoiled plasmids can enter other cells more efficiently, and plasmids have been used extensively in recombinant DNA technology to introduce specific traits again. Okay, so that means class, by manipulation of the plasmid, okay, or the plasmid DNA in vitro, okay, the specific genes can be introduced into a new cell. That's why we have that recombinant DNA technology by the use of plasmid. So you have molecular cloning, we have gene therapy, we have recombinant DNA technology, which are, of course, applications of these plasmids. Okay, so this is, I know you have this in the previous discussion and also in your laboratory. So we have here, for example, okay, we have blunt ends, and of course, okay, we insert this specific sequences, and this one, okay, will be inserted to the plasmid. Okay, and these are the different types of vectors, class. Plasmids, bacteriophages or viruses, bacterial artificial chromosomes, yeast artificial chromosomes, and mammalian artificial chromosomes. And these are the characteristics of a cloning vector. So number one, it must be small in size, so it can be easily manipulated. It must be self-replicating inside the host cell. It must possess restriction site for restriction endonucleases, because you need to take note, class, that the restriction endonucleases or the restriction enzymes will cleave the plasmids, and the same enzymes will be used to cleave, okay, the DNA of another cell or another host. Okay, what is important in having that same restriction enzymes is to make sure that you are really making sure that the fragments or the new genes that you are inserting to the plasmid is, of course, will fit, okay, the sequence coming from the source DNA. Okay? Next one, introduction of donor DNA fragment must not interfere with replication property of the vector. Okay? Must not interfere with the replication property of the vector. And cloning vectors are used for transferring fragments of foreign DNA into a suitable host. They play an important role in selecting recombinants from non-recombinants. Question, how plasmids are used in genetic engineering? Again, we have, I have mentioned gene therapy, I have in antibiotic resistance, I have mentioned molecular cloning, okay? I have also mentioned about, what did I mention earlier? I forgot. Okay? Recombinant DNA technology. Okay? And of course, we can make use of that generally in genetic engineering. So the foreign DNA fragment is inserted into the plasmid. The foreign DNA fragment is inserted into the plasmid, and the recombinant DNA molecule is transformed into the recipient cell. The foreign DNA fragment is inserted into the plasmid, and the recombinant DNA molecule is transformed into the recipient cell. So that's how the plasmid can, of course, be genetically engineered so that the recipient cell can contain the characteristic that was incorporated in the plasmid. So the transformed cells are selected by the antibiotic resistance of the used plasmid. Okay, just like this one, class. Let me just zoom this out. Okay, so this is a type of molecular cloning. Okay, so the foreign DNA and plasmid, okay, let me just change this one. So the foreign DNA and the plasmid are cut with the same restriction enzyme. This is what I'm telling you, okay? Because when both the plasmid class and the DNA insert will be cut by the same restriction enzyme, what will happen is they will have complementary ends. So if they have complementary ends, it will facilitate the ligation process, okay, where the DNA insert is inserted into the plasmid vector, okay, and the complementary ends again can base pair with each other. So meaning to say, efficiency of ligation can be increased. Okay, that's again very important that you are using the same restriction enzyme. So going back, the foreign DNA and plasmids are cut with the same restriction enzyme which recognizes a particular sequence of DNA, and that is what we call as restriction site. So the restriction site class occurs only once in the plasmid and is located within the LAC-Z gene, a gene necessary for metabolizing lactose. This one class. Okay, so in this illustration, okay, we have the restriction site here. Okay, we have the restriction site. Okay, so we have one restriction site in this example. Okay, and of course, that restriction site, okay, is located in this gene, the LAC-Z gene, okay, in metabolizing lactose. Another one, the restriction enzyme creates sticky ends, okay, sticky ends, just like in this illustration, okay, that allow the foreign DNA and cloning vector to anneal, okay, to anneal, to stick to each other, and an enzyme called ligase glues the annealed fragments together. I hope you still remember ligase. Okay, so this one class, this is the foreign DNA, this is the plasmid, and of course, restriction enzymes, okay, are used to cut both the foreign DNA and the plasmid, right? So that they will have complementary ends. And what enzyme now will stick the fragments together from the foreign DNA and from the plasmid? Okay, it's the ligase. Okay, and the ligated cloning vector is transformed into a bacterial host chain that is ampicillin sensitive and is missing the LAC-Z gene from its genome. Okay, so you have here. And another thing, that one, it will be multiplied, okay, and you will have such colonies, okay, with the foreign insert. Okay, I'll explain that one later. I'll give specific examples of that one. Okay, especially some of the terms that you might encounter. Okay, so just like this one, we have the E. coli, we have the plasmid. So the plasmid will be isolated, so it's here. The foreign DNA, okay, example, this gene containing the resistance to antibiotics. So you will isolate that also, this one. And then of course, cut the plasmid with enzyme. So the plasmid is cut here. That's what you can see here. Okay, and cut the cell's DNA with the same enzyme. Take note again, I have mentioned same enzyme to get this gene of interest. Let's say antibiotic resistance. And you will combine the plasmid and the foreign DNA containing that, okay, antibiotic resistance. Okay, and what will join them together is the ligase. So you will now have the full circle, the recombinant DNA plasmid containing again the gene of interest. Okay, you understand, class? Okay, for example, let me just illustrate here. In the laboratory, you have non-selective agar, for example. Let me just enlarge that. You have this non-selective media, this one. Let's say this is nutrient agar. Nutrient agar is non-selective, right? And then another one, you use another agar, but this time you incorporated antibiotics. Let's say you incorporated kanamycin. So this is now the kanamycin agar. And then you divided it into half. This one also. Okay? Next one, in this nutrient agar, in the left-hand side, there is no plasmid containing the resistant genes to antibiotic. Let's say kanamycin. In this one, there is a plasmid containing the resistant genes. And then you strict the bacteria. Since this is a nutrient agar class, which of the two sides will grow? Non-selective. Na is non-selective. Regardless of the genes of these bacteria, may it be containing plasmid or not, they will still grow. That's why you can see colonies in this area, okay, and colonies also in the area with a plasmid. However, class, if you use another agar, which is incorporated with an antibiotic, which is kanamycin, okay, especially, for example, what we are talking here is the kanamycin-resistant gene, for example. So, in a nutrient agar, both of them will grow. However, if you strict class, okay, in this part, the one without still, with no plasmid, and you strict the one with plasmid containing the kanamycin resistant, okay, which will grow class, the left-hand side or the right-hand side. In this case, you cannot see growth on the left-hand side, but you can see growth on the right-hand side. Because, again, this plasmid, okay, contain the gene necessary for resistance to what antimicrobial? Okay, kanamycin. So that's a simple activity which you can differentiate, okay, the type of bacterium and the resistance to antibiotic. Okay, another one. Okay, class, for example, I will illustrate here, okay, 3 bacterium. So you have bacteria 1, let me get another color. This is bacteria 1, bacteria 2, and bacteria 3. Bear with me. Okay, another one, we have this chromosome, reverse chromosome. And then another one, okay, you have the plasmids here. Plasmids. So let me enlarge the plasmid. Let's say this is the plasmid. Okay, so they're double-stranded, right? So bear with me, class. This is an art class. Okay, next one. This is plasmid also. And this one, sorry for that. This one doesn't contain plasmid. Okay, it's only the bacteria 1 and 2 containing the plasmid. Okay. Okay, next one. Okay, this plasmids class, number 3, this one. Okay, these plasmids class were incorporated, sorry for that, were incorporated with resistant genes. So let's say you insert a resistant gene here, same resistant gene. Let's say that resistant gene is resistance to ampicillin. This one resistant to ampicillin. Another thing, you want the plasmid to be resistant to tetracycline. So, you inserted another gene there. And again, this one contains resistant to what antimicrobial I mentioned. Okay, tetracycline. This one also, we have tetracycline, right? And then let's say I have a Petri dish here. And then this Petri dish class, okay, this Petri dish contain ampicillin and tetracycline. That Petri dish, that agar. And then you see, let's say, 3 colonies. Okay, colonies. Let me enlarge that one. Okay, numbered 1, 2, and 3. If this agar class contains ampicillin and tetracycline, which of the 3 bacteria will grow in this agar? Will one grow, yes or no? Yes, one will grow because it has this ampicillin-resistant gene. How about number 2? Yes, it will grow. How about number 3? This one. Of course, it doesn't have plasmid, so it will not grow, okay, in this agar, right? However, what if the term insertional inactivation happens. Okay, let me write that one. Insertional activation happens. When you say insertional activation, class, you are inserting another gene or regulatory element in the gene which is previously found to carry a particular characteristic. For example, class, this is a tetracycline resistant gene, right? So both the bacteria, okay, have this tetracycline resistant gene. When you say insertional activation, class, you insert another gene or regulatory element in that part of the gene. And what will happen is this one will be inactivated. That's why it's what you call as insertional inactivation. Meaning to say that previously gene which was inserted, which is found to be having a characteristic, okay, in exhibiting tetracycline resistance will be inactivated. Sorry, I misspelled it. That is insertional inactivation. Inactivation. Okay? What will happen if you have here a culture medium and then another culture medium? Okay? This one, let's say we separate the ampicillin and the tetracycline. Okay? And then you strict the first bacteria, sorry for that, this one, you strict the first bacteria, this one, second and third, same with that one, first, second, and third. Okay, 1, 2, 3, this one, 1, 2, and 3. Okay, what will happen, class? Okay, will bacterium 1 grow in ampicillin? Does it have ampicillin resistant gene? Yes, of course, so it will grow. How about bacterium number 2? Will it grow in ampicillin agar? Yes. How about number 3? Of course, there is no plasmid, so it will not grow. How about in tetracycline? Previously, both of them, okay, grow in the ampicillin and tetracycline agar, right? How about if insertional inactivation happens? Insertional activation was conducted in bacteria number 1, bacterium number 1. Will bacteria or bacterium number 1 grow in tetracycline agar because the gene for tetracycline resistance is inactivated? Of course, this one will not grow. How about bacterium number 2? It will grow, and bacterium number 3, it will not grow. So that is the term insertional activation. And I hope when you read those things in your books, okay, you will not be confused already. Okay, so in the laboratory, that's how we manipulate the bacteria to differentiate them. And of course, if we want to study a particular characteristic such as antibiotic resistance, we can perform these things that I have illustrated to you a while ago. Okay, any question, class, about the plasmid? Okay, thank you so much. So I hope you read your books, because that is the key to passing the exams. And also for your examination, class, you have restriction enzymes, you have plasmids, and lastly, we still have one topic left. Okay, we have blotting techniques. Okay, southern blot, northern blot, and western blot. So all of those things will be included in your exam. Okay, and also on Monday, May 6, okay, 5 p.m. to 7 p.m., we will be having our quiz, 2 quizzes, okay, for the topics restriction enzymes and plasmids. So for the types of quizzes, you have multiple choice, you have matching type, and you have also true or false. So again and again, please read your books. Do not just rely on my presentation. Do not just rely on the things that I am saying. Always read your books so that you can have an in-depth knowledge of the topics that you are learning. Okay? Any question? Okay. Thank you very much. God bless. God bless, class.